are made up of proteins, lipids and carbohydrates. So here we have to see how protein is synthesized in our cell. So before starting with the protein synthesis, let us revise the structure of cell. This is the outermost covering of the cell, that is cell wall. Inside it cytoplasm is there. Then different organelles are present and nucleus is also present. Nucleus is very much important part of the cell. And last year we have seen that inside the nucleus chromosomes are present. And those chromosomes are made up of DNA. Now these DNAs, from this DNA mRNA is formed. We know that DNA means what? Deoxyribonucleic acid. It is double stranded structure. And RNA, that is ribonucleic acids, it is single stranded structure. Now in the nucleus, from this DNA mRNA is formed. DNA is present inside the nucleus. RNA is present in the cytoplasm. So from this DNA mRNA is formed. This procedure is called transcription. It is a part of protein synthesis. After formation of this messenger RNA, this messenger RNA comes in the cytoplasm. Means the process of transcription takes place inside the nucleus, and this mRNA comes out in the cytoplasm, and from this mRNA protein is synthesized, and this procedure is called as translocation. So the process of transcription and translocation together is called as central dogma. What it is called? Central dogma. Now in short, we have seen that transcription takes place inside the nucleus, translocation takes place in the cytoplasm, and these two procedures are called as central dogma, and in this way protein synthesis takes place. Now in detail we are going to see the process of transcription and translocation. So let us see the structure of DNA first. Last year we have already learned the structure of DNA. It is a double stranded structure, two strands are there. It is a ladder like structure and it is twisted around itself, and this DNA is made up of 4 nitrogenous bases. They are adenine, thymine, guanine and cytosine. 4 nitrogenous bases are present along with deoxyribose sugar and phosphate molecules. So in this way the structure of DNA is there, and always adenine paired with thymine and guanine paired with cytosine. Now at the time of protein synthesis, some part of DNA gets unwound. For example, if this part of DNA is to be unwound, this part is unwound and this remaining twisted, the remaining part is twisted, only this much part is unwound. So here you can see A is paired with T, C is paired with G, T is paired with A, G is paired with C. So pairing always like this, A always with T and G always paired with C. So in this way pairing is there and it is unwound and this new strand is formed. New strand is formed and this new strand is nothing but the messenger RNA. So but one important thing we have to keep it in mind, in case of DNA thymine is present and instead of thymine in RNA uracil is present. What? Uracil. Uracil is present instead of thymine in case of mRNA. So whenever this strand is forming, A is paired with U. You can see here this A is also paired with U. Everywhere, wherever this thymine is there in case of DNA, here you can see uracil in case of messenger RNA. Now this strand is formed. Now, this is called as messenger RNA. Now it will come out of the nucleus. And you can see this mRNA. This is the mRNA strand, single stranded structure. Here you can see GACU. These nitrogenous bases you can see. Now one important, they are forming pairs. Three pairs. Three nitrogenous bases form a pair, that is called as codon. That is called as codon. You can see this is say first codon, this is second, this is third. Such type of codons they are forming. Triplet codon. Three are there in each codon. In each pair, 3 nitrogenous bases are there. That's why they are called as triplet codon. Now always starting codon is AGC. AGC means what? Wherever this triplet codon AGC is there, from that place protein synthesis starts, and that's why it is called as starting codon. That's why it is called as starting codon. And at the end codons are there. End codons are there. So wherever end codons are there and wherever starting codons are there, from protein synthesis start, and at end codons it will stop. In this way protein synthesis takes place. Now you have seen that these codons, triplet codons are there, and now here transfer RNA comes with the anticodons, means those are the pairs of these 3 codons. For example, pair of A is U, pair of G is C, pair of C is G. So these 3 anticodons it has brought, and with this one amino acid, specific type of amino acid also it has brought. Whenever it has brought this anticodons, means the codons, pairing codons you can say, complementary codons you can say. These complementary codons they are bringing, and specific amino acid it has brought. Now this amino acid will keep here only, and this transfer tRNA will move forward. Then next tRNA will come with the anticodons. For example, here codon is UGA, so anticodons will be AGU. So for these 3 anticodons, specific type of amino acid also will be there. Let us consider it is AA1. Again it will move forward, and this amino acid will keep here only. Then next transfer RNA, tRNA will come having anticodons according to the specific codons, and according to this codon, specific type of amino acid it will bring. Let us consider AA2 is the amino acid. In the same way again next will also come, and with their anticodons and specific type of amino acid. Similarly, such amino acids, every anticodon, every transfer RNA will bring such type of amino acids, and those amino acids will join with each other to form a chain, and this chain of amino acid is called proteins. So as and when they will leave these amino acids here, these transfer RNA leaves their place and move forward. So only this chain will form here, and this is the protein. Protein is nothing but a chain of amino acids. So this chain of amino acids is formed here, and after the formation of this chain, this messenger RNA vanishes and it will finish, and therefore we have already here we have seen this is the temporary one. This is the temporary one, and that's why it is called as. So in this way this chain of amino acids is formed and this messenger RNA